Okay, so this audio lecture will cover the information in Chapter 11, looking at physical control. So again, with this unit, we're looking at preventing the growth of microbes. And for physical control, we're talking about non-living surfaces. And so, in our journeys, we can actually see ways in which companies have incorporated, the science into their different products so that it inhibits microbial growth. And so one of the things we'll talk about is heavy metals like copper. And so if you go into some of the airports, you'll actually see that the water bubblers, which are probably taped off and not allowed to be used now, might be embedded with copper. And so that has this antimicrobial property. And so one of the times I was at the Atlanta airport, I noticed this, so I snapped a picture of it. So when we look at microbial controls, they fall into three different categories: physical agents, chemical agents, and mechanical removal methods. Okay, and we'll look at each one of these. You should definitely know an example for each level of cleaning. So sterilization, disinfectant, antiseptic, and decontamination. This figure is one of the nice figures, so if you're going to print one figure for this chapter, this would be the figure because it really nicely summarizes up all the different agents, methods that we'll end up talking about. I won't talk about every single one. I will highlight some of the key ones that are traditionally used and such. Okay. Now, with the controls of microbes, there's some terminology, and again, I kind of refer to this as levels of clean. So sterilization is where we're going to be devoid of life, so there's no living cells, material, microbes on the surface. So do realize that sometimes when in newspaper articles or lay articles, they tend to overuse the word sterilization because if there's living cells, technically it's not sterile, but the idea is that we have no microbes present. And another thing to keep in mind with sterilization, really the benchmark here is no endospores. That becomes relevant later on when we talk about the different organisms and their ability to survive different cleaning methods. So with sterilization, we're looking to see that there's no endospores present, which means any of the vegetative cells and viruses would be inactivated so that they wouldn't be able to cause infection. With disinfection and antiseptic, these are looking at removing any of the vegetative cells. For disinfectant, it's a non-living surface. Antiseptic is the term we'd use for a chemical, a solution that would be used on a living tissue. Okay. And then with decontamination, what we're doing is we're trying to decrease the number of microorganisms so it's safe enough to handle. So in no way is it disinfected or sterilized. It's just decontaminated. And so in most cases, if we think about like a household setting, decontamination is what we're actually trying to achieve. We're just making sure it's safe to handle, and also keeping in mind what that room is used for. So obviously your kitchen should probably be cleaner because that's where your food prep's happening than your bathroom, because making some assumptions, but you're probably not eating in your bathroom. That may not always be the case, but you're not preparing food in your bathroom, and so just having decontamination so it's safe to handle is sufficient. In your kitchen, you're probably not performing surgery, so the level of sterilization is not necessary. I know tons of cleaning products will tell you, like, we need to sterilize everything, but the reality is, if you're preparing food on the surface and then you're cooking it, the cooking process will actually kill off most of the microbes. And not that I'm saying don't clean your cutting boards, but sterilizing your cutting boards isn't necessarily the level of clean you need. Definitely disinfect it, but even if you're just getting a decontamination level, because you're going to be cooking that food afterwards, that shouldn't make a difference unless you're contaminating it during the cooking process. So keep in mind that different organisms have different levels of resistance. And so in this table, if we look at endospores being more resistant than vegetative cells, if we look at different methods such as using heat with moisture or irradiation or using something like the sporacine glutaraldehyde solution that we use in our lab. You can see how endospores are extremely resistant, where vegetative cells are more sensitive. I just want to point out that sporacine liquid, the vegetative cells, you need 10 minutes of contact, and if you review your lab syllabi, we say 2 minutes. So again, in the lab, we're not really sterilizing the bench tops, or even, I think it would be hard to say that we're actually even disinfecting the bench tops, because we're not allowing for 10 minutes of contact time. We're probably more getting, decontamination again. So it's safe to handle. You're going to minimize the chance of contamination. Now, we had talked about the idea of microbial death. I didn't use the specific term microbial death, but we had talked about how we can have something that is cidal. So in our example here with this figure, we have something that's germicidal, so it would kill germs. So we can see a dramatic decrease in the number of bacteria that are found when exposed to this germicidal soap. You'll also notice in the yellow line that we have this non-germicidal soap, or germostatic soap, okay? And so you can see the levels kind of go up and down, but it kind of remains the same. So they're not increasing in numbers dramatically, but they're not decreasing, they're not dying, and they're kind of halted, right? Like we had said, how they freeze, they're not able to reproduce or replicate. And so there's really two ways to achieve microbial death. It's where we actually go and we kill the microbe. Again, that would be cidal, or we could have something that's static, and that's where it's not able to reproduce or replicate if we're talking about a virus. There's a number of factors that affect microbial death. So the length of time that something is exposed to that method. So obviously the longer we expose the surface to a specific method, we're going to be more likely to reach sterilization. Also, what's known as the load. So if we have a high amount of microorganisms or a high load, it's going to take longer in order to achieve microbial death or to get sterilization. Where if it's a low load, it's the surface that's being cleaned regularly, and we don't have a lot of buildup, we would be able to achieve sterilization sooner. And I would say that's where, thinking about the micro lab, we have a number of labs going through. You're cleaning bench tops before and after, so there should be a low load of microbes on it, and so you're going to be, even if you're just doing 2 minutes of contact time, you're going to get a higher level of clean. You're going to be more likely to achieve microbial death. Okay. The type of microorganism that's present, so in panel C, we see how we have endospores or spores, and we have vegetative cells. And so if it's just vegetative cells that are present, you're going to be more likely to achieve microbial death in a shorter time than if you have endospores, because endospores are going to be more resistant. In D, we see the different types of agents. So if we have something that's microcidal, it's going to kill the microbe. So you're going to see a dramatic decrease or achieving of microbial death much quicker. With something that's microbial static, you have to continue to give it or have that cleaning product, that method present in order to eventually, like what we see with the antibiotics, eventually potentially kill it, okay? But you're going to have this long period where it's just not capable of growth. But if you stop the cleaning method, if you stop whatever method you're using to achieve microbial death, if you stop that, it can start growing again. So there's several different mechanisms of action. Try not, some of these are kind of redundant to the antibiotics, but these are unique to the physical control. Okay, so mechanisms of action would be disruption of the cell membrane. So just like with the polymyxin, it has a very detergent-like appearance. So we can actually have detergents, surfactants, and they're going to disrupt. And so as you can see in the figure, when the detergents are applied, like soap, you can see how it gets incorporated into the plasma membrane, and then it actually disrupts it, so then the cell is not able to be selectively permeable. Now, the mechanism of action is affecting protein function. So keep in mind that proteins have to be folded in order to have function. And so we can denature those. That can be done by heat, change in pH, exposure to heavy metals. And so what happens is the protein's now in a different shape, as we see in the figure, or denatured. And so it's not able to function. And when proteins can't function, they can't do what they need to do in the cell, and obviously protein's important, and so the cell is not able to metabolize or to function, and so again, this is where we can get something that's either static or cidal, depending upon how severe the treatment is. So usually, sometimes what happens is you get coagulation, so we get these proteins clumping together. So that's achieved through heat, alcohol, acids, and phenols. So next we're going to look at some of this physical agents. So keep in mind that we have heat, but we have dry heat versus moist heat. And then we also have radiation that we'll talk about. So with heat, there's a difference whether there's moist heat or dry heat. Moist heat meaning that there's water molecules at a high concentration available. And so if we think about, where people like to retire, they always say they want to retire somewhere where there's dry heat. And that's because, dry heat, 100 degrees doesn't feel like 100 degrees if there's a high humidity, right? So they know what's going on. The science behind that is those water molecules. If you have moist heat, those water molecules can do work. They're moving around rapidly, and they can, bombard the cell and disrupt membranes and disrupt the proteins being able to function. And so this can have a negative effect to it. If you have dry heat, you don't have those water molecules there to do the work, and so you have to have a much hotter temperature. So keep in mind that water boils at 100 degrees Celsius. If we look at these higher temperatures, like 121 degrees Celsius, we can achieve sterilization in 15 minutes. That same temperature, if it's a dry heat, you would have to sterilize, you'd have to expose it to that heat for 600 minutes. So 10 hours in order to get sterilization. So obviously with the water molecules present and the moist heat, a much shorter time, which is obviously if you're looking at trying to get surgical equipment or something like that, you wouldn't want to be like, oh wait, wait 10 hours, then we'll get your equipment. You want something much quicker. Now the way to avoid these water molecules from evaporating away is by putting them into a closed system. And so this actually can create pressure, and that pressure actually allows you to get the water up to the temperature of 121 degrees Celsius. So the autoclave, which you see there in the figure, is the technique or the equipment that's used in order to create this steam under pressure system, very much like a pressure cooker, where you can cook foods in a quicker time period than if you just cooked them in like your oven. So the amount of pressure that's the autoclaves usually set it is at 15 psi's, or pressures per square inch. Now when we look at dry heat, one of the common techniques that's used in the lab is incineration with your Bunsen burner, where you incinerate your inoculation loop. Keeping in mind that flame, that if you have that nice blue cone on your flame, that tip of the flame is around 600 degrees Celsius, so it's extremely hot. So you're able to, light anything that's on that inoculating loop on fire, so it gets incinerated. Large-scale incineration can be done, so that's done with biohazardous material, and so that's where you have like companies like Sterilization and other companies that will come in, but some hospitals and medical facilities will actually have on-site incineration, and so that they can incinerate any of that biohazardous material so that those potential pathogens aren't exposed to other people. So then looking at radiation, there's two different forms of radiation. There's ionizing radiation. This is where an electron gets ejected, and this causes ions to form. And so this can cause DNA breakage. And so if there's breaks in the DNA, then the DNA is not able to replicate. There's also non-ionizing radiation. This is where atoms get excited, but we don't have the electron being ejected. So we don't get these ions being formed, but we do get abnormal DNA bonds, okay? These abnormal DNA bonds can lead to mutations, okay? So it's important to keep in mind that with these what happens to the DNA is different between ionizing radiation and non-ionizing radiation. Okay, and then we also have UV radiation, but that's not able to penetrate very well past surfaces, so that you wouldn't be able to get deep tissues effects. Okay, so for controlling the growth of microbes, it's ionizing radiation that is looked at, and there's several different applications that are utilized. So in food products, medical products, so medical products like saline solutions, and also some material like suture material that can't be put into an autoclave. So there's some material that is heat sensitive, and so you need a way in order to treat it so that there aren't microbes on it. So ionizing radiation is utilized. Also, it's been looked at for food product, so keep in mind food spoils. We have to refrigerate it because it will spoil. So the whole idea of refrigerating it is to slow microbial growth. And so if we could ionize your food, there wouldn't be microbes on it, and then the microbes wouldn't be able to spoil. Benefit of ionizing radiation is that you could have something like, your chicken breast or your steak, and it would actually be able to penetrate through. Okay, really quick. So it's basically you put something in the conveyor belt and it continues on, and it gets passed under this light, and the ionizing, which is an ionizing radiator. Okay, I actually attended a conference on food microbiology, and they actually talked about the use of ionizing radiation. And for those meats that you don't want to have to ground up, like a steak, this is really promising because, again, it's able to make sure that there's no potential pathogens in it, and that the food isn't damaged like you would, obviously if you autoclaved your food, it wouldn't come out the same as it went in. So UV radiation, it is also utilized, but what can happen is you can get mutations induced because you get mismatching. So you get these pyrimidine dimers forming, and it doesn't have the same penetration power, so it's not able to get, it's only like a surface treatment. So the applications of UV radiations that are used is when we want to disinfect something. So like a hospital room, an operating room. We're also seeing UV radiation used in schools, and dental offices. If you're in the micro lab, there's a hood, and that actually has a UV light on it, so that hood area inside will actually get treated. It's also used for drinking water, so you can pass in some towns, for their town drinking water, city drinking water, they will pass it through UV lights to kill off any organisms that would be potentially in there. This link is for a hospital in Massachusetts that is using UV robots. So the UV robots will go into the patient's room and kind of scan the room, expose the surfaces to UV light. Again, it doesn't penetrate in, so there's a little bit of prep of opening drawers and making sure those contact surfaces are exposed to the UV light so that it will decontaminate, disinfect those surfaces. So next we're going to jump over to mechanical removal methods, so looking at filtration. So filtration can range from, in the lab setting or in the home setting. So you think about your HV system, your HV system. is going to pass the air in your house through filters, right? So if you have the joy of owning a home, or your parents have ever made you change the furnace filter, you'll know that there's this filter there, and all the air passes through, and it gets filtered through the filter, the membrane. The filter is going to have holes in it, and so if you go to, a home improvement store, or even like a Walmart or Target, you can buy different filters, and you can buy filters with different hole sizes. You can also buy filters with charges, and so if you have a filter that has a charge to it, you can potentially remove viruses from the air in your home, which sounds great, but the reality is most people are getting exposed to the virus because they're sitting close to each other in the room and exchanging air before it goes through the filter system. Okay, so I usually say save your money. You don't need the highest, bestest, supposedly by the company, filter, because again, most people are being exposed because they're sitting in the same room as someone potentially before they're even symptomatic. Okay. In a lab setting, they can have HEPA filters. So these are high efficient particle air filtration systems. And again, those, if it's a situation where you have a room that has negative pressure, so it actually flows the air out of the room, these potentially could remove pathogens, viruses, before someone is able to breathe them in, so the recirculation. And this is obviously a big focus looking at school systems and hospitals with COVID, making sure that their HV systems are removing the air so that people aren't being exposed to what people are breathing. But obviously their systems are a little different than your at-home system. Another way filtration is used is for liquids. So here in this figure, we see how we have this media, and we want to make sure that we don't have any, bacteria present in it, and so we're going to filter it. So it goes through this membrane. You can see there's little holes in it, so it allows the liquid through. Now, keep in mind, most of these filters do not eliminate virus. So the fact that it's a sterilized fluid, it's not truly sterile because there could be viruses present. Okay. Also, sometimes media fluids can't be filtered because we might actually filter out some of the larger proteins that we actually want in the media. Okay. So there's pros and cons. The pro would be that with filtration, there's no heat, so there's no risk of thermal damage. All right, so next we're going to look at the chemical agents, and we're going to focus mostly on liquids. So in the healthcare setting, a number of different chemical agents are utilized. So I'm not saying memorize this chart, but maybe think about what you're using in, when you go to the doctor's office, or when you're doing your clinicals. And one of the things we always have to keep in mind is the level of activity and potential toxicity, because people could be exposed, are being exposed to these chemicals. So ideally we want to use something with as low toxicity as possible, but with the highest activity needed. Okay, and again, how clean something has to be in a doctor's office versus a surgical wing is very different. And so we want to keep in mind that, in the surgical wing, we might want something that's sporicidal, but in the, doctor's office, something that is just a little weaker on the scale, something that is, has some activity, that's partially bactericidal might be sufficient. We don't necessarily need to go to the level of sterilization. We also want to keep in mind that concentration and time matter. So the higher the concentration, the less time you need to expose the surface to that. So I like to just point out ethyl alcohol, your hand sanitizer. So it's not all at 70%. So some of it's down in the 60% level. And so if we want to kill something like E. coli, we need 2 minutes contact time. So think about the hand sanitizer you put on your hand. I know it's in a gel form, so there's still some residual amount, even though like the gel has kind of evaporated. But you're probably not getting 2 minutes contact time. So usually it's evaporated off after 30 seconds. Whenever I've had students try this out. So after 30 seconds, they'll be like, yeah, good. And then you might have some carryover on that, but whether you're getting that 2 to 10 minute contact time is very questionable. Okay. One of the good things with COVID is that it has a membrane, which is made up of like lipids. So they're a little more sensitive to the ethanol. The guidelines say at least 60%. Personally, I'd want something above 70%. If I'm using solely hand sanitizer, better to wash your hands with soap and water. Okay, so the question might be, well, okay, well, Dr. Schreiner, what chemical would you prefer to use? And I would say hydrogen peroxide. So you can see hydrogen peroxide at 3%, which you can get at your grocery store, or any kind of store that would sell cleaning products. 3%. Normally, maybe not right now, but normally, and then within 13 seconds of contact time, you have things like Staphylococcus aureus, which would require 10 minutes of 70% ethanol. It's killed, so very quick. I do recognize that hydrogen peroxide has a bleaching activity. So again, you got to kind of weigh what surfaces you're using. But if you're using something like a cutting board, I don't know about you, but I'm okay with it getting a little bleached in color. So we're just going to look at a couple of the different cleaning methods. First are the phenols. So your Lysols are a phenol-based product, and so these at high concentrations can actually be cidal, so they can actually be toxic to you also, so we have to keep that in mind with toxicity. At lower concentrations, what they're going to do is inactivate critical enzyme systems. So this is that protein function piece, and so that the enzymes can't catalyze reactions, so the cell wouldn't be able to survive that long term. Okay, trichlorosin is a chemical that's put into a lot of the hand soaps, with recommendations by the FDA that these antimicrobial chemicals are actually should be removed from hand soap. So some of your newer hand soap may not have it, but definitely there was a period about 2 years and back where we saw a lot of these chemicals being added in, and in a traditional setting, just the detergents alone are sufficient. We don't have to add additional because all we're doing is starting to breed resistance. There's also a lot of people who are actually sensitive to trichlorosin, and so it can cause skin lesions and such, and so obviously that's not beneficial because now you could potentially introduce microbes into those wounds. I already had mentioned hydrogen peroxide, but hydrogen peroxide relies on certain enzymes to be present in order to handle the free radicals that are being formed. And so catalase is one of the enzymes you'll look at in the lab, and so we actually use hydrogen peroxide to test whether the bacteria has catalase present. At high concentrations or long exposure, it can be harmful to your tissues, but it is a product that can be used as an antiseptic or disinfectant. Okay, and it can be bacterial, viral, and fungicidal. And at high concentrations, above 3%, so you have to buy those. For a hospital setting that wants hydrogen peroxide that's sporicidal, that kills endospores. They would be purchasing that directly from the chemical company, and you can get that as high as 60%, but also 30%. And you need special license, so you can't just call up the company and be like, give me some 60% hydrogen peroxide, I'll just dilute it out, and it'll be a whole lot cheaper. It's really expensive. And again, you need special license in order to get that. And then, as I just was mentioning with detergents, detergents act as surfactants, and so again, they have a very phospholipid-like look to them, and these are cidal because they disrupt the membranes. Heavy metals can also be utilized, like I had mentioned with the copper, but silver and gold can also be utilized. So here's basically, a Kirby Bower where they put some of these heavy metals, they put silver and gold onto this plate, and you can see how you have a zone of inhibition, so the bacteria can't grow. So silver is more common because it's cheaper. So you can have silver creams that are used for burn victims or individuals with poor wound healing. And the silver it has antimicrobial properties, and so it's harder for the microbes to grow. It kills many of the microbes, so it allows the patient to have a higher success rate with recovering from the burns or from the wounds. Okay. You can also buy like bandages that have silver embedded into the bandages. Very expensive, so it's about like $15 for a 10-pack. But again, if you are having problems with wound healing, then it's probably worth that extra cost to get it. You can also buy toothbrushes that have silver embedded, and there's a lot of in the equestrian world with horses, they actually have water buckets that have silver embedded into it, because in some regions where, especially if they have the water buckets outside, issues of salmonella growing in the water. This is an issue more, I've heard of cases in like California where horses will come down with salmonella infections, and they trace it back to the water buckets. So again, the idea is by kind of not lining but embedding the plastic of the bucket with the silver, it actually inhibits the growth. Horses and livestock, they're also prone to getting like a fungal growth on their legs, and so they also make these like socks that have silver embedded into the fibers. And then we have glutaraldehyde, or aldehydes, glutaraldehyde, formaldehyde, or formalin. So these actually cross-linking between proteins. So you can see here how these proteins have been cross-linked, and so they're not going to be able to rearrange, and so it kind of locks the cell into position. So they would be static, so they don't die, but they can't move their proteins. So they're kind of frozen. So formalin is used as a fixative for tissues, for like biopsies and such. Glutaraldehyde does have some toxicity to it, so again, the choice of applications, you got to be mindful about that. And so one of the things that glutaraldehyde is used is on breathing tubes, so they will actually treat with very low concentrations with the breathing tube to make sure there's no microbes growing, because obviously we don't want to introduce a contaminated breathing tube to a patient. Traditionally, patients don't tend to be on having a breathing tube long term, so it's kind of a risk analysis, right? That potential exposure to the chemical where it could cross-link their own proteins, it's worth that because they need to breathe, and because the breathing tube is sensitive to heat, can't be filtered, you're kind of limited in what cleaning methods you can use. So this is a list of some of the different antimicrobial products that are kind of common household use. So again, you might want to kind of look at, which cleaning products you're using in your house, and what are the chemical agents, and what category does it fall into. Lastly, I do just want to mention that, we talk about sterilization being no endospores present, which means everything below endospores there, you can see it's the second microbe on there. Everything below that would be killed by those methods. With disinfecting, we are talking about killing off the vegetative cells, and so this can, depending upon the method, kind of falls in the range where we might be able to, we'd get all those vegetative protozoans, but obviously protozoans can form cysts, and so those would be able to survive those methods. Okay, you will notice that there is something above the endospores. So we got the prion diseases. These are these protein-based diseases, tend to be neurological diseases. And so traditional sterilization methods like using the autoclave are not sufficient. So because prion diseases are extremely interesting, because again, it's a protein it's spread through proteins, which just doesn't make sense. There's no nucleic acids there. We used to think there were these viruses. They're not. And so the area that there is a concern of transmission is with neurosurgery. So equipment that is used for neurosurgery has to go through additional cleaning methods. So usually they use 3 methods that would individually lead to sterilization in hopes that the purons, these infectious agents, are removed. Up until recently, there were no, quick tests to do this, but there was a there was an incident in the New England area where a piece of surgical equipment was used for a patient who ended up having this puron disease. They didn't know it at the time. The patient died, and then when they did the autopsy, and the only way to do an autopsy for puron disease is actually to send the body to the CDC. And so when it was confirmed that the individual actually had this puron disease, they then went back to look at who had used that piece of equipment afterwards, and they are following those individuals. So there's about 30 individuals that this piece of equipment was used. Because some of the surgical equipment is extremely expensive, it gets rented out. And so it's not just individuals that were treated at that hospital with that equipment. There's a number of hospitals in the New England area that use that piece of equipment between the time it potentially has this infectious agent on it to the time that it was identified of the patient actually having this puron disease. And so with that, there was obviously a push to come up with testing testing that can be done on a live individual, where they actually will take spinal fluid and look for these infectious agents in the spinal fluid. And also with that, they also came up with ways in which they can test for puron diseases on the surgical equipment. But again, the gold standard is to do kind of like 3 sterilization methods, usually like an autoclave method, and then a gas method, and then they will also do a cleaning solution method. And again, all of these individually should reach sterilization, but with the puron diseases, because they're extremely resistant, and obviously a hospital doesn't want to use this piece of equipment and then potentially infect their patients, because it has virtually 100% fatality within 6 months of showing symptoms. So definitely something you don't want to get. And again, normally the transmission with these are through interaction with contaminated like meat or brain material. And so, if you had surgical procedures done, you're probably fine. It's very rare, but when someone does get it, it is fatal. So, all right, so that concludes the lecture for chapter 11 on physical control. If you have any questions, please email, and we will discuss it in class.